Okay, everyone, we're going to get started. Hello, welcome to Genome Webinars. I'm Ed Winnick, Editorial Director for Genome Web Daily News, and I'll be your moderator today. Today's webinar is entitled Optimization of NGS Library Preparation, Low Inputs and Fast Streamlined Workflows. The sponsor of this webinar is New England Biolabs. Our panelists today include Cynthia Hendrickson of the Hudson Alpha Institute for Biotechnology, Momo Vujicic of the Los Alamos National Laboratory Genome Center, and Daniela Munafo of New England Biolabs. You may type in a question at any time during the webinar. You can do this through the GoToWebinar control panel, which usually appears on the right side of your screen. If the panel is minimized, just click on the small red box with the white arrow to expand the control panel. We will ask the panelists questions after the presentations have concluded. We're going to start with Cynthia Hendrickson from the Hudson Alpha Institute for Biotechnology. Dr. Hendrickson, please go ahead.
Thank you. As Ed mentioned, I'm a part of the Genomic Services Lab at Hudson Alpha Institute, and today I'm going to present some of our data using the NEBNext Ultra DNA sample prep kit in conjunction with Exome Enrichment for simple, low input, and low cost exome sequencing. Next slide, please. The Hudson Alpha Institute for Biotechnology is a nonprofit research center located in Huntsville, Alabama. As such, the Genomic Services Lab within Hudson Alpha is a cost recovery next-generation sequencing provider that not only supports researchers within the Institute, but customers worldwide. We offer the full spectrum of next-generation sequencing sample preparation, including whole genome and target-enriched DNA library prep, as well as a variety of RNA and epigenetic sample preparations. And we offer sequencing on Illumina's HiSeq 2000, 2500, and MySeq, as well as sequencing on the Ion Torrent and 454 platforms. Next slide, please. So one of our most popular services is exome sequencing, and we typically use around 2.5 micrograms of DNA and perform a standard DNA library preparation. In this method, DNA end repair, A-tailing, Y-adapter ligation, and PCR amplification are performed in discrete steps, each followed by a bead cleanup. This method is time-consuming. It requires multiple aliquots of beads and necessitates multiple transfers of each sample, which can result in sample loss and increases the number of plates or tubes required. After library preparation, we typically use an Immunogen SeqCap EZ human exome. However, we have modified the protocol so that we can pull 2 libraries together per exome capture. This literally cuts the cost of exome enrichment in half. However, the method does require indexing the libraries prior to pulling and enrichment as well for enrichment, as well as using adapter blocking oligos that are specific to these indexed adapters. However, Immunogen does provide the sequences for index blocking oligos specific to Illumina adapters as part of their protocol. Next slide, please. So instead of using our usual DNA library prep, New England Biolabs invited us to try their new NEBNext Ultra DNA library prep method upstream of our exome enrichment. In this method, end repair and A-tailing occur in a single step, after which a unique cleavable hairpin adapter, which is more stable than Y adapters, is ligated. After ligation, a user enzyme mix is added which cleaves a U in the hairpin, generating the more traditional Y-shaped adapters. The pulse allows amplification of the library. Next, please. So our current method takes about 3.5 hours. This new method actually removes about an hour from the process. This end prep with end repair and A-tailing occurring in one step takes 60 minutes, 15 minutes for ligation of the adapters, in which the adapters and ligation master mix are simply added to the current reaction. After 15 minutes, user is again simply added to the reaction. And after an additional 15 minutes of incubation, there is a single bead cleanup. There is the option to do a bead-based size selection in the protocol, but we don't typically use size selection for exome enrichment, so we kept with a simple cleanup. After that is PCR enrichment, followed by one last cleanup. As a result, not only did this reduce an hour of time, but also cut the number of bead cleanups and sample transfer steps in half. Next slide, please. So to compare the methods, since we anticipate a reduction in sample loss with the NEBNext Ultra, we decided to push it, instead of going in with our regular 2.5 micrograms, to go in with only 500 nanograms. But compare that to our current method with our normal inputs. Using both methods, we perform 6 cycles of PCR amplification after and repair a tailing and adapter ligation. In the case of our current method, we use Kappa's Hi-Fi Hot Start, and for the NEBNext Ultra, we use their new high-fidelity master mix that's included in the kit. Seeing the results of the library, we found that we got less than our inputs, which we normally see in library yields with the current method, with 1 microgram to 1.2 micrograms. And with the NEBNext Ultra, we actually got a little bit more than we started with 649 and 529 nanograms. After taking some of the material for QC, we went ahead and pulled 2 libraries per capture, 500 nanograms each with our current method, and 450 nanograms each with the NEBNext Ultra samples. The captures were performed as we normally do. We performed 15 cycles of PCR afterwards, and then we found that the pool of 2 libraries, again, we had more material coming out with the NEBNext Ultra at 2.5 micrograms compared to our current method, which was 1.5 micrograms. So going on to the next slide. For sequencing, we put them on a HiSeq 2000. We generated paired in 100 reads, and you'll notice that there is a difference in the number of reads generated with the current method and the NEBNext Ultra. That's simply because we are a service provider, so whenever we try new protocols, we squeeze the samples in wherever we can. The space available for the sample in this case was a little bit less of the lane than we had for our standard current method samples. However, aligned pairs and aligned sequence were comparable, with over 99% of the reads aligning to the genome using WA and aligning to G19. The depth of coverage obviously reflects the number of reads generated. Quality filtering metrics were comparable, and when it came to the number of duplicate reads generated, with our current method, we saw 14% and 13%, which is on the high end of our normal. And with the 500 nanograms NEBNext Ultra samples, we saw 8% and 11%, which is on the low end of our normal. Going on to the next slide. So because we used 20% of our normal input, we took a closer look comparing the sequencing results. And just looking at the distribution of reads across the chromosomes, you see that although, of course, there are a few less reads with the 500 nanogram samples, that they do basically mirror themselves in distribution across the chromosomes. More importantly, looking at the actual amount of sequence that was on target, plus or minus the fragment size around 200 base pairs, we see that 87% of the sequence was on target with our normal method, and 86% with the 500 nanograms, which is within error randomness. This was important for us to look at because our primary concern using a new library prep method for target enrichment is that the adapter blocking oligos are perfectly compatible. Seeing that the number of off-target reads were comparable between the 2 samples, we know that the blocking oligos were in fact equally as efficient with blocking the NEB adapters in comparison to normal Tllumina adapters. On to the next slide. So taking a little deeper look, we also looked at the distribution of reads across the chromosomes. This is just randomly looking at the first arm of chromosome 1, and again you can see that the distribution of reads is comparable. Given that the 500 nanogram samples prepared with the NEBNext Ultra kit performed as well as the 2.5 micrograms prepared with our normal method, we decided to push this method further and go back to the same DNA stocks, the same 2 individuals that we used for the 500 nanogram prep, and go in with 100 nanograms just to see what would happen. So going on to the next slide. So comparing, again, in this case it's the same individual, 100 nanograms and 500 nanograms. Again, you see that the distribution of reads across the chromosomes are comparable, and in addition, the percent on target. With, again, the 500 nanogram gave us 86% sequence on target. The 100 nanogram samples gave us 88% sequence on target. And then on to the next slide. And again, zooming in and looking within the chromosome, you can see, again, the distributions are similar, that, although it was a lower input, we're not seeing regions falling out or, any unusual PCR bias that we wouldn't want in our samples. So on to the next slide. So since it is the same individual, we were able to delve deeper. And so we looked at the specific variants that were identified using 500 nanograms of input versus 100 nanograms of input. As you can see, based on the number of variants per chromosome, again, the patterns were very similar. In general, the number of variants detected at 100 nanograms were a little bit less, but you can see that in some cases, for example, chromosomes 16 and 19, the number of variants in the 100 nanogram sample was a little bit higher. Specifically, a little over almost 59,000 variants were found with the 500 nanogram and 51,000 with the 100 nanogram, showing only a 13% loss in the number of variants detected with a significant reduction in DNA input. And then on to the next slide. And so then specifically looking at the single nucleotide polymorphisms, again, we found that basically we were identifying the same ones that'll match to the DB-SNP 135 database. You can see that over 10,000 were shared between the two samples, whereas almost equal amount were discovered in one of the sequencing runs versus the other. So the depth of coverage just wasn't optimal. And finally, on the last slide. So we found that we were able to get excellent results with the NEBNext Ultra DNA sample prep kit. We were able to use DNA inputs far lower than we had dared use before. We obtained greater yields post-library prep and generated similar sequencing results. In addition, the library prep was simpler and faster than our current method and required less beads and sample transfers. So I would just like to finally acknowledge everyone in the Genomic Services Lab at Hudson Alpha that had some part in generating this data. In particular, Nupresh Prasad, who did most of the bioinformatic analysis, and also thanks to New England Biolabs for allowing us to try their new kit and share the results with you today. In particular, Pingfeng Liu, who gave her support in helping us incorporate this method into our exome enrichment protocol. Thanks very much.
Okay, thank you, Dr. Hendrickson. Just a reminder to everybody, if you have a question, please type it in the question box. It should be on the right-hand side of your screen. We will pool the questions and ask the panelists at the end of the webinar. Thank you very much. Next up is Momo Vujicic of the Los Alamos National Laboratory Genome Center. Dr. Vujicic, please go ahead.
Hi, everyone. Go ahead and switch to the next slide. So I will be talking about our research and development projects that involved the NEBNext Ultra library prep methods that we actually tested here before they were released. And we've obtained a lot of data, and we're very, very happy with this library prep method, and I'd like to tell you all about it. So I work in the Genome Center at Los Alamos National Lab in northern New Mexico, and this is we have a variety of projects that are mostly R&D projects that are collaborative with universities and industry and other government agencies. And so you can see here the variety of projects that we do. We have a large sequencing facility that has all of the modern NGS platforms, and we also have a vast computational analysis division that can handle any data for transcriptomics or metagenomics or genome assembly or analysis or anything like that. Next slide, please. So we do two types of sequencing that requires very high level of even coverage for bacterial genomes. So we do genome sequencing of bacteria, and here what we do is we prepare short insert shotgun libraries, and we generally use Tllumina for sequencing these, and then we assemble the reads into contigs. So these are continuous or contiguous stretches of DNA that can be assembled from reads that cover the genome. And you can see that in the upper left side. And on the upper right side panel, you can see that this approach of trying to cover the entire genome is not perfect, in that if you look at the y-axis, which shows the coverage of the genome, you can see that there are many gaps along the genome for which the sequence cannot be obtained using many of the library prep kits that we've used in the past. We also do a genome finishing. We're well known for that here at Los Alamos. And so for that, there's a special type of a library that we prepare. These are called long insert paired end libraries. They're basically 8 to 10 kb fragments that we sequence only the ends of, so we can put these contigs that we generated in the genome sequencing together into a circular chromosome that bacteria have. And I will not talk about that much, except that we used to use 454 sequencing for that a lot. Now we're switching to PacBio and Illumina sequencing. Next slide, please. So the NEBNext Ultra, so we had a very high throughput bacterial genome sequencing and assembly and finishing going on until the budget cuts were introduced. Now we're just waiting for more budget approvals. But this is our standard process for sequencing bacterial genomes now using the NEBNext Ultra library prep process. So we tested several commercially available kits, and this one actually showed the best overall performance in pretty much every possible way. So we can use very small amounts of input DNA. It is very inexpensive. It is very easy to perform. It's fully automatable, and it produces data that are as good, if not better, than any other kit available. And so here on the left side, you can see the steps involved, but it's fairly simple. It's basically two consecutive reactions in the same tube, followed by one purification using Ampere XP beads, and then PCR amplification and PCR cleanup. That's all there is to the prep, basically. And so what we did, you can see on the right side, experimental parameters. What we did for all the kits that we tested, we applied them to 3 different bacteria that have three different GC contents. So Bacillus anthracis at 31, E. coli at 50, and a Burkholderia strain at 67% GC content. We also varied the input DNA amount at 10 and 100 nanograms for this kit, but we had to go up to a microgram with certain other kits. And then we also tested the ability of betaine to improve the GC coverage during PCR. And then we varied the library fragment sizes. And if you look at the workflow, it's basically DNA quantification. We use qubit for that. It's very accurate and sufficient. We determined the DNA integrity of the genomic DNA using eGels, 1% EX. That's a 10-minute eGel run. It's all precast, and it just takes 10 minutes. DNA shearing, we always perform in Covaris E210. This gives us very consistent results, and it's just the best process that we found. Size selection, we performed either with Ampere XP beads or with eGels, and I will only show you, I think, the data for Ampere XP beads because they perform very well. They're fully automatable. They're easy. Library quantification, again, we've performed with qubit. Library sizing, we've performed with Vinalizer 2100. However, we are now convinced that that is an unnecessary step because the size selection is so reproducible that we can simply assume that the size is always the same and simply quantify the library and generate the molar concentration of the library. So then we performed the normalization of all libraries down to 2 nanomolar for Illumina sequencing. We sequence either on HiSeq or MiSeq, depending on the project, and analyze the data. And you can see on the left side that the total amount of time is about 4.5 hours for library prep. Next slide, please. So the first part that was important to us was to select the fragment sizes that were of interest to us. And so since we normally sequence on the Illumina platform using 2x150 base pair sequencing, so these are paired end sequencing from both ends of the fragment, we wanted fragments that roughly are of 270 base pair length, such that the 250 base pair reads will overlap slightly and will give us better quality of data towards the end of the reads, and then we can assemble full 270 base pair fragments that are then very useful for both read mapping and assembly. And so you can see here that we were going for optimization of DNA shearing using 200 to enrich for 270 to 300 base pair insert size. And so we simply dilute each DNA sample to 100 nanograms or 10 nanograms in 55 microliters. And these are the cycling conditions of the Covaris E210. This is extremely similar to what NEB recommends, except that we varied the time and we observed that 100 seconds was the best for our requirements, but that's something that anyone can implement whatever their process requires. And so you can see here the size distribution of genomic bacterial DNA after 100 seconds, that it's very nice, and this is very reproducible. I could show you 10 of these and they would look exactly the same. So that's very nice that Covaris works reproducibly well. Next slide, please. So you can see now, once we go through the process of NEBNext Ultra, here's one example library. We call it POA-16 for our limb system, but it's a Burkholderia genomic DNA, so it's 2 large chromosomes. And here's our prepared library. So when we add about 270 base pair insert to the 120 roughly base pairs of the adapters, we arrive at roughly 400 base pairs of the library fragment size average, plus minus. So this distribution is exactly what we were looking for. There are no adapter dimers. There are no primer dimers. It's very clean. It sequences beautifully. And so here's the summary of what we did here and what we obtained. So 100 nanograms starting material of this specific Burkholderia strain. We performed ampere size selection, 8 PCR cycles with bedding. Final concentration of the library is 14.9 nanograms per microliter, or 52.6 nanomolar. So that's way, way more concentrated than necessary. So it's possible to pull back on the number of PCR cycles. Average fragment length is 429 base pairs, and we sequenced this on HiSeq. You can see in the right panel the insert size histogram, and this is actual based on sequencing, and it's very, very accurately representing what we observed experimentally in the lab. So the insert size is basically perfect for our needs. Go ahead and switch to the next slide, please. So when we look at the actual sequencing data, this is exactly the same library when sequenced. So Burkholderia bacteria have 2 chromosomes, about 4 megabase pairs each. You can see here that we had low coverage overall, but the coverage is very even. It's around 50-fold coverage. And you may or may not be able to see that, but in the upper right-hand side of each of the coverage plots, there are some stats, and they're showing that there are very few gaps and very few base pairs that are not actually mapped from the reads obtained. And so the first chromosome, there's only 11 base pairs out of 4 million base pairs that are not covered by this sequencing. And in the second chromosome, there's 143 base pairs that are not covered. So the coverage is very even, and it covers almost 100% of the genome. Next slide, please. So here's another example. It's a sample POA-15. It's the same exact strain, but we started here with 10 nanograms of the genomic DNA input. Ampere size selection again. We had to go to 12 PCR cycles, and you can see the concentration is still of the library very high, 11 nanograms per microliter, so 42 nanomolar, very high, the same exact average library fragment size, sequenced it on HiSeq, and the insert size histogram on the right side shows exactly the same size distribution of the insert sizes, very, very consistent. Please go to the next slide. And if you look at the genome coverage, again, 50x coverage, and we have pretty much identical coverage, and we only have 7 base pairs of gap in the first chromosome and 129 of the second, so that's basically identical results obtained at 10 nanograms of input DNA, which is really impressive because no matter what the sample is, if you can pretty much quantify it with anything, you can sequence it using this method. Next slide, please. Here's another example of a different Burkholderia that now goes to a higher coverage level. So this is 150x coverage of both chromosomes. The size distribution is beautiful. The evenness of coverage is absolutely beautiful. There are no large gaps. There are very few gaps. And so this is very suitable for either read mapping or assembly. Next slide, please. Here's now a Bacillus anthracis Strand strain, and again showing that with a low GC organism we can achieve the same results. You can see here the average fragment size is 400 base pairs. We started with 100 nanograms of input and only performed 8 PCR cycles and still obtained 72 nanomolar library concentration, no primer dimers, no adapter dimers, very clean and nice. Next, please. And here's the coverage plot. And so you can see that the plasmid PX01 is covered very well to a very high level, of course. The genome is also covered to a high level, and you can see that this was a rapidly replicating genome because at the origin of replication on the left and right side of the graph, we have twice as much DNA as in the middle because the DNA was being replicated from the origin of replication away from it. But again, the coverage is very, very even, and we only have 7 base pairs out of 4.6 million that are not covered by this sequencing method. Next, please. So just wanted to mention to us, this is very important, these 2 slides here. The slide on the left is we simply applied this method that we developed to our single-cell genomics efforts that we have here. So we sequence a lot of single cells from complex microbiomes. And so here we sequenced an MDA amplified product from a single bacterial cell processed with the NEBNext Ultra. There's a bit of an adapter dimer, that's the 126, and there's a bit of a primer dimer at 83. However, that's really negligent and can be completely excluded computationally. But you can see that the size distribution of the library is beautiful. It sequenced beautifully and gave us beautiful data from single cells. And then another important aspect of this kit is how robust it is. So the very first time that I trained a student who didn't have any experience with these kits, she was able to reproduce absolutely beautiful data with exactly the same. You can see the library size distribution is exactly the same. There are no adapter dimers. It's just worked beautifully and it's sequenced beautifully. So that's a very important aspect, that this method works every single time on any given day in anyone's hands, and that's one of the reasons we love it. Next slide, please. So we have done this extensive project with comparing different kits on different bacteria and perform different computational analyses to try to both assemble, to map genomes and assemble them. And this is just a quick graph showing how many contigs and scaffolds we obtained when we combined Tllumina data that I showed you with PacBio data for the long insert libraries, and then all paths assembly. You can see that we, for Burkholderias, it's particularly challenging to assemble those genomes because they're high GC content. So we obtained quite good results. And for example, Bacillus anthracis on the right side, we obtained very good results. There are very few contigs and very few scaffolds. In fact, it was fully sequenced. The full genome was sequenced. Next slide, please. Well, that's all I have. So I'd like to thank very much the NEB team that helped us work through this process. It was very easy and nice to work with them. Also, we have the LANL wet lab team and the LANL computational team on the right side. And for more information about any of this, you can contact me or Patrick Chain, who is my colleague that does all the computational analyses. And I have a couple of pictures from the beautiful Southwest.
Okay, thank you, Dr. Vujicic. Our final panelist today is Daniela Manafo from New England Biolabs. Please go ahead, Dr. Manafo.
Good afternoon, everyone. Today I'm switching gears. In the talk I'm going to present directional RNA sequencing from low input amounts. Next slide, please. So as you are probably aware, standard RNA-seq workflows are very laborious and time-consuming because they involve numerous enzymatic and cleanup steps that leads to significant sample loss and reduction in library yield. Therefore, making these workflows not compatible with low inputs, neither suitable for automation. And in most cases, what is even more important, they lose the information about strand orientation. Next slide, please. So standard or directional RNA sequencing had been shown to substantially enhance the value of an RNA-seq experiment. by providing information of which strand of the double-strand DNA gets transcribed, helping to identify sense and antisense transcripts that are transcribed from overlapping genes, and helping to demarcate exact boundaries of adjacent genes transcribed from different strands, and more accurately measure gene expression or transcript abundance. Next slide, please. So NEB is introducing a new tool for directional RNA sequencing. This is the new NEBNext Ultra Directional RNA-seq library prep for Tllumina. And this kit has incorporated all the advantages and the improvements that of the NEBNext Ultra DNA kit that had been nicely highlighted by Cynthia and Momo. And we incorporate those improvements, and we upstream the reactions up front from RNA isolation, fragmentation, and priming, as well as the double-strand cDNA synthesis, making the workflow combining enzymatic reaction steps. This workflow is fast, simple, and more streamlined. And what is more important, the number of cleanup steps have been reduced only to 2. So it makes the workflow fast. It can be done involving only 30 minutes of hand-on time and reducing the total time to 5 hours compared with the 2 days that it used to take a traditional standard workflow. And moreover, since the number of steps transferring sample has been reduced, it's making it compatible with low input amount. So this workflow is compatible with as low as 100 nanogram of total RNA and as low as 10 nanogram of purified mRNA or ribosomal depleted RNA. Next slide, please. So going in more detail into the workflow, as I mentioned, the workflow is compatible with ribosomal depleted RNA as well as total RNA. So if your star material is total RNA, this workflow can be combined with the NEBNext poly-A mRNA magnetic isolation module to isolate the mRNA on magnetic beads. And in one step, the mRNA can be eluted from the beads, at the same time fragmenting, and the random primers get annealed. So we are performing this elution step in a buffer that contains magnesium. So by heating the sample, these 3 reactions takes place all at once in the same step. There's no need to purify the RNA fragments, so therefore there is a reduction in sample loss. So at the end of this step, the RNA fragments are annealed with a random primer and ready to perform first-strand cDNA synthesis. For this step, we are including a new RT enzyme provided by NEB that is an MMLULB RT enzyme, RNAH- that can increase the yield of the cDNA synthesized. For second-strand cDNA synthesis, there's no need to clean up a step before first strand going into second-strand cDNA synthesis. During this step, the buffer that we're using for second-strand contains uracil, and so the sample, the second-strand DNA, gets labeled with the UTP. We are using this method with consistent marking of the second strand with the UTP and posterior excision of this strand after adaptor ligation. This method for directional RNA sequencing has been highlighted in the literature as one of the best methods. And if you are curious to learn more about the method, you could refer to the paper that we highlight at the bottom of this slide. This technology has been developed by Max Planck and is now incorporated for New England Biolab. And so, as I said, second-strand cDNA contains uracil. There is an end-repair tailing step that allows to create blend end and then over TA over hands of the double-strand cDNA. Again, all the enzymatic improvements done for the NEBNext Ultra DNA kit have been incorporated, so I'm not going to go over Cynthia and Momo nicely highlight the improvements done in the downstream steps. So this after second strand DNA synthesis and repair a tailing get done, there's no need to clean up and adapter ligation occur. We are using harpinine adapters developed by NEB that also contains uracil. Therefore, there is, before going into PCR, there is an user excision step. During this step, the loop adapters open, as well as the second strand that was marked with uracil gets degraded. So, so only one strand DNA goes into PCR, and this strand during PCR amplification gets differentially tagged at the 3' and 5' end with P5 and P7, preserving strand orientation information, and this is what it makes this workflow directional. And also during PCR, barcodes can be introduced, multiplexing the library. Next slide, please. So if we look at the library performance, what I'm showing here in Figure 1 and Figure 2 is the typical library profile that we obtain. In Figure 1, we are looking at mRNA libraries made from 1 microgram of universal human references total RNA. And since we have optimized site selection, you can see the library has a very nice narrow profile with a main peak at around 270 base pairs. The library is very clean, no adapter dimer in the primer line dimer form. And the typical yield for 1 microgram total RNA library is around 500 nanogram. So it's a half a microgram typical library yield. On the right side of the figure, I'm showing that it's also possible to go as low as 100 nanogram of total RNA, as is shown in the bioanalyzer traces. In Figure 2, we are looking at ribosomal depleted RNA library. And this library was made with 80 nanograms of ribosomal depleted RNA. And again, the same nice narrow size distribution profile with a main peak around 270 base pair. And a library yield around 600 nanograms. On the right side of this figure, I'm showing that it's possible to go as low as 10 nanograms of ribosomal depleted or even 1 nanogram. And I'm not even showing in this library, but we have also made libraries with 100 picogram of ribosomal depleted. But we see some inconsistency at such a low level. So for the 1 nanogram, for instance, we see a little bit of adapter dimer, but that can be prevented by diluted adapters or maybe just performing an additional cleanup step after the library is done. We have fully characterized the 10 nanogram ribosomal depleted library, and in later slides, I'm going to provide some more data. Next slide, please. So on the screen, what you're looking at is that the quality of the sequencing data generated by Directional Ultra RNA-seq kit. For this purpose, we made libraries with 1 microgram of total RNA, universal human references, total RNA, as well as ribosomal depleted RNA. We, for comparison purpose, we choose to make libraries with the Tllumina 2.6 stranded mRNA kit. We sequence those libraries on a single enriched 50 base pair reads on a Tllumina instrument, 1 sample per line, and we generate roughly 30 million of reads per sample. On Figure 1, what we see is we are looking at the percentage of mapped reads when those reads are mapped with the RefSeq, and we see a high percentage, more than 90% of the reads mapped with the references, either in mRNA libraries or ribosomal depleted libraries than with the RNA Ultra kit. On Figure 2, we are looking at how well those transcripts were covered. So this graph on the axis, we are looking at the whole transcriptome, how well it's covered from the 5' end to the 3' end. We are looking at the relative distance across the transcripts from the 5' end to the 3' end. On the y-axis, we are looking at the relative read coverage. And so we will see is that there's a nice even coverage distribution on libraries that mRNA libraries as well as the ribosomal depleted libraries than with the NEBNext Ultra kit. And as is expected, the mRNA libraries had a better coverage at the 3' end since these are single-end reads. On figure 3, we actually were looking, we wanted to see how much ribosomal RNA was left after using the NEBNext mRNA magnetic isolation module. And so what we see in this figure is that the NEBNext libraries had very low percentage of ribosomal RNA left after the enrichment. It's less than 1%. And this is very nice because we get much more information about the, our RNA-seq experiment, mapping roughly 80% of the RNA with the coding regions. Next slide, please. So we wonder how robust this workflow was, and so for this purpose we investigate how well technical replicates correlate. And so in this slide I'm showing correlations between technical replicates and correlations between different inputs. So if we look at the graph that is in the upper side of this slide, we are looking at NEBNext Ultra mRNA libraries made with from 1 microgram of total RNA, 2 different samples, and we're looking at the FPKM and how well this FPKM correlates between samples, and you can see there's a nice correlation with a coefficient correlation of a 0.99. And when we look at how well the transcript correlate between different input amounts, we also observed that there was a nice correlation of libraries made from 10 nanogram of ribosomal depleted RNA comparing with the 100 nanogram ribosomal depleted RNA libraries. And next slide, please. Next slide is just highlighting for the technical replicate graph. I'm highlighting, I'm zooming in the low expressed transcripts, and so you can see better that even at the low expressed transcripts also correlate very well. And what it was nice to see is that we saw that the libraries at low input, the 10 nanogram ribosomal depleted libraries, we found comparable transcripts numbers with the high input libraries, meaning that libraries retain complexity even at low inputs. Next slide, please. So when we look at the strand specificity, if you are doing a non-directional workflow, as is expected, 50% of the reads will randomly mapped to what the annotated strand. That in this bar graph, the annotated strand is shown in blue as a sense, and 50% of the reads will map to the antisense strand, shown in the graph in red. So that is if you are doing a non-directional workflow where there's lost information of a strand orientation. For NEBNext Ultra Directional Kit, what we see is that 98% of the reads map with the expected or the sense strand. They're making this method highly strand specific. We observe that actinomycin D is required, and the reasons for this is that an intrinsic activity that has the RT enzymes, the reverse transcriptase enzymes during first strand DNA synthesis can not only extend over RNA templates, but they can also extend over newly synthesized first-strand cDNA. And if this happens, a second strand is made during first-strand cDNA and does not get labeled with DUTP. Well, actinomycin D inhibits this DNA-dependent DNA polymerase activity, therefore increases strand specificity. Next slide, please. So to illustrate the directionality and the stranded of the NEBNext kit, I am sharing with you one slide that our collaborators from Children's Hospital has generated. This data is done in E. coli. What we're looking here is E. coli transcriptome then using the NEBNext Ultra Directional Kit. And what you can see is how nicely the reads mapped on the annotated strand, and there is very low background on the opposite strand. And what I'm highlighting here on the squares is the transcription start sites. And what you can see, it's overlapping mRNA transcripts at the transcription start sites. And to prove that this is not an artifact of the method, next slide, please. What our collaborators did is they expressed the P19 protein in E. coli. P19 protein can bind to double-strand RNA, short RNAs, 21 nucleotides, and only double-strand RNA. And so what we see in the P19 libraries, that these double-strand RNAs were pulled down, and then directional libraries were made by using a direct ligation of adapters to the RNA. This is a different method to do a strand-specific libraries, but for short RNAs. And so you can see how nicely the P19 libraries overlay in the areas where there are the mRNA transcripts can overlay at the transcription start site. So this is confirming by a different strand-specific methods that the areas that we see overlap are actually real and not artifact of the NEBNext Ultra method. Next slide, please. So finally, in summary, I present today a new tool that NEB is providing for directional RNA sequencing with a stream protocol, just 5 hours total time, that can accommodate low inputs amount and provide higher library yields. As I showed by the replicate assays, the method is very robust, highly strand-specific, and which we think it's very nice that it can retain diversity of the library even at low input. Last slide, please. Finally, I want to acknowledge all the people that it's been involved in the development of this kit, and thank you for listening.
Okay, thank you, Dr. Manafo. Thank you, Dr. Manafo. As a reminder to attendees, if you have a question, please type it into the question field in the GoToWebinar control panel. We have about 10 minutes to ask the panelists questions and get their answers. So I'm going to start with Dr. Hendrickson. Our first question was, does full spectrum include RNA genome applications?
Yes. We do
okay, thank
both
you. Another one of our... I'm sorry, go ahead.
Oh, no problem. We do both non-directional and directional RNA as well as small RNA.
Okay, thank you. One of the questioners wanted to know, how do you account for the increased post-capture yield with the NEBNext, given you are inputting about the same amount of template for PCR?
I would assume it would have to be reflective of the number of fragments that actually get both adapter ligated on them. So we're quantifying using qubit or bioanalysis that reflects, the amount of DNA, but not the amount that's PCR amplifiable.
Okay, and actually a question for both you and Momo. Attendees would like to know what has been your cost per sample?
Well, in my case, we were given a kit to try, so there wasn't one.
Well, this is Momo from. You can actually now purchase this Ultra kit from NEB, and I believe the cost per sample is $24 for the entire kit, and that does not include the Covaris tubes, but those are $2.50 a piece, and those would have to be used for many other kits as well. So. Yeah, the cost is very, very low, and then we use biomech robots to perform the automated library prep, so there's very little labor involved in the process as well.
Okay, thank you. Cynthia, also, have you tried the NEB method on buccal or FFP?
No, not yet.
Momo, sorry. One of our attendees wanted to know if you've tried another shearing method, and if so, what?
We have not with this kit. Not with this kit. We have tried to use enzymatic methods, and they just were not as consistent as this one. So this is just very, very reproducible.
Okay, thank you. And how many putative PCR duplications in 100 versus 10 nanograms? That was from Momo.
Oh,
or Cynthia.
I don't know. We didn't do that analysis.
Cynthia?
In our case, we actually did have an increase in duplicates because we increased the PCR cycles, but we realized that that was a mistake, and we will not increase PCR cycles in the future and just keep it at the 6 cycles.
Okay, what, this is for any of the panelists, what is the cost of the NEB kits compared to the Illumina?
Hello. I would refer the people to just browse our website.
Okay, thank you, Daniela. How about the cost difference between the NEBNext system and other traditional prep methods? Has anyone looked into that?
I believe, this is Momo, I believe that the TruSeq kits cost $60 per library, I believe, but I'm not 100% sure. But I know that they're significantly, and NEBNext is significantly less expensive, and so our lab manager, that's what my lab manager told me. So I don't know what the actual cost is, but I'm sure that can be easily found out online.
Okay, thank you, Momo. Also, we have had a few attendees asking about if you perform, this is for Momo, if you perform sequencing on the ion torrent or proton using this library prep method.
We did. So we used the NEBNext Ultra for ion torrent, not for the proton, but we used a version of the kit that included enzymatic DNA fragmentation, and that's the part we did not like too much. And so we switched to Kovaris shearing, and now we're getting great results. We're getting very, very consistent results on the ion torrent, and so that's our standard method now for ion torrent as well, and we just got the proton, so it'll be for the proton as well.
Okay, thank you. And Daniela, can the NEBNext kit be used for other next-gen sequencing platforms like the Solid?
One requirement for make the workflow directional is that the adapters cannot be blend-end. It has to have the harping shape or either the Y shape. So directional workflows are not compatible with the blend-end double-strand adapters like are used in Solid. But if someone just can go around with the shape of the adapters, it will be compatible. That's the only requirement for compatibility for the directional. However, I didn't went over the talk. NEB also offer an NEBNext Ultra kit for RNA that is not directional, and that will be more compatible with blend and double-strand adapters.
Okay, and sticking with you, Dr. Manafo, somebody want to know how will these adapters perform with products randomly amplified, say at a size between 150 to 400 base pairs?
Can you repeat that question, please?
Sure. The question was, how will these adapters perform with products randomly amplified, say at a size between 150 to 400 base pairs?
They perform very well. So, for example, for DNA, We have a isolation chart, so the DNA fragments can be isolated in different range, and actually in the ranges that you just mentioned. And ligation performs fine with these adapters in different insert sizes.
Okay, great. This is for any of the panelists. Have you used this method with mammalian RNA isolated from FFPE tissues?
Yes, I have made libraries with FFPE sample from human, and library was performed fine. However, we are working on making it more upstream, and it does not, something not related to the workflow itself. It's FFPE samples can be very variable in terms of quality, and we are putting a big effort to understand what are the limitations that makes sometimes fail depending on how the FFPE RNA has been isolated. But we have successfully made FFPE samples using the NEBNext Ultra kit.
Okay, thank you. Also, one of the attendees noted that magnetic beads are used to enrich eukaryotic RNA, but what do you suggest to do with bacterial prokaryotic total RNA?
At the moment, we do not offer any solutions for ribosomal depletion from prokaryotic. There are products available out there in the market, and what we have seen is variability in terms of a lot of variability in terms of performance of this product. So some of the data that I share with you was done with one of the best methods, and it performs very well. And we have also tried the kit with other methods that do not work as well in terms of depleting ribosomal RNA, and we see a little bit less percentage of mapped reads. But at the moment, we do not offer anything for ribosomal depletion.
I'd like to add something. This is Momo. So we do a lot of transcriptomic analyses of bacteria and eukaryotes and mixed samples, and we have had very good results with ribo0 reagents from Epicenter. And that's all we use nowadays, and we expect good results, and we get them every time. Ribo0 is specifically specific for removing ribosomal RNA, of course.
Okay, thank you, Momo. We have come to the end of the webinar. We would like to thank our panelists, Cynthia Hendrickson, Momo Vucic, Daniela Manafo, and our sponsor, New England Biolabs. If we didn't have time to get to your questions, we will try to have the panelists answer them directly afterward. Also, please look out for 2 post-webinar polls from the sponsor before you log out. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to attendees. Thank you for joining us for this Genome webinar, and we hope you participate again.